Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum and welcome back to our cell biology, genetics and evolution class. We started the topic of recombination in bacteria, and in the previous few videos we discussed the process of transformation. That is how a bacteria gets transformed from one strain to another, and that was a famous example of type 2R transformation into type 3S. So this is what we discussed in the previous videos. Now our parent topic is recombination, that how genetic material from one bacterium gets transferred into another bacterium. And as we saw in the first video, there are three mechanisms: transformation. The next one, or the second mechanism, is conjugation. Conjugation is basically a mating process, and it involves bacteria. Since our discussion is about bacteria, how genetic material gets transferred from one bacterium to another. So a mating process involving bacteria. What happens in conjugation is that genetic material from one bacterium gets transferred to another bacterium through physical contact. That is, the two bacteria in question, they are in physical contact, and genetic material is transferred from one bacterium to another. And you might be wondering how it is possible that there is a mating between the two bacteria and then genetic material gets transferred. So this contact, as I said, that it involves physical contact between the two bacteria. Now this physical contact, suppose these are two bacteria, involves a special tube made of protein which connects the two bacteria. And this tube, it acts as a channel, a passage, a gateway between the two bacteria, and this particular tube is known as F pilus or sex pilus. So this is the tube. It is a conduit, it is a passage, it is a channel which makes possible the transfer of DNA from one bacterium to another. The question is, where does this tube come from? How it is formed and how it connects the two bacteria? This F pilus or sex pilus can be made or can be synthesized or constructed, whatever you call it, by the bacteria which contain the F factor or fertility factor. So that's why it is known as F pilus, F factor or fertility factor. So it involves this particular piece of DNA, and the cell which contains this F factor or fertility factor is known as F plus. So F plus cell, it contains the F factor or fertility factor. The other cell which lacks this particular F factor is known as F minus. So it lacks the F factor. Now this mating or physical contact between the two bacteria involves F plus and F minus. That is, a cell which contains this particular piece of DNA or F factor and the other cell which lacks. So these two cells come in contact, and how it is done and what is this F factor? F factor is what we call in genetic terms an episome. Okay, so what is an episome? As I just said that F plus cell becomes F plus because of an episome. The episome is genetic material, and as I said earlier, it is mostly DNA. DNA of what? DNA of phage. And this particular piece of DNA, it exists independently of the DNA of the bacteria in which it is present. Independently. And bacteria replicates it when the bacteria replicates its own DNA. So this particular piece of DNA is also replicated. The episome that we are talking about, which is F factor or fertility factor, and this one contains about 19 genes which is present in the bacteria, and this particular F factor makes possible the synthesis of F pilus or sex pilus. So this F factor, it imparts the bacteria the ability to, or it confers the ability to conjugate upon its host cell, that is in which it is present. So this is a circular piece of DNA containing about 19 genes, and it is responsible for the synthesis of F pilus or sex pilus, through which conjugation takes place or the transfer of DNA takes place. Now, here is the overall process of conjugation. Now we have two bacteria, and this is the other one. This particular bacteria is F plus and this one is F minus. Before going further, let me tell you something, that during this process of transfer of DNA, no recombination takes place, as you will see shortly. This is just to introduce you the concept of conjugation, because there are certain variations in this conjugation process. Although the process that we have just started the discussion of uh, does not result in recombination, other conjugation processes do result in recombination. So you will get an idea that what is conjugation and what happens during conjugation and how conjugation can bring about recombination. So these are two cells, F plus and F minus. Now this is the chromosome of the bacteria. We are going to draw double circles to indicate the double-stranded DNA. And this is the other bacterium with its double-stranded DNA. And we are going to change the color slightly. So this is the episome present in the F plus cell. So this is episome or the F factor, and this is DNA of both the bacteria, DNA of bacteria, this one and this one. Now what happens that this F factor or episome, it initiates the synthesis of F pilus or sex pilus. So what happens that an F pilus is synthesized. This is F pilus, F plus cells with F pilus. Now it has formed an F pilus, and this is the other cell as we have just drawn, F minus. Now it contains its own DNA, and it has a circular piece of DNA. Now in the next diagram, what happens that this F pilus, it connects the two cells. So the two cells are connected and the transfer of the DNA begins, or the transfer of this particular F factor begin. Now the two cells are connected by virtue of the F pilus, and what happens that this F factor or the fertility factors, its transfer is started from F plus cell to F minus cell. So let's draw that. So this is F factor and one of its strand gets transferred, and as it enters the recipient cell, the synthesis of new strand begins. So this continues, and if we draw another figure, so after a certain time, certain length of time, the transfer of the F factor is completed. However, this F pilus is a fragile one, and in the meanwhile it's broken, and but the transfer of the F factor or fertility factor has taken place, and the overall situation after the transfer of the F factor is somewhat like this as I am drawing it now. And this is the donor cell, the original F plus cell. Now the cell which was previously F minus has also become an F plus cell because it has received the F factor or the fertility factors. Now both the cells are F plus cell, and this one which was previously an F minus, now it can also initiate conjugation, and now it can also synthesize the F pilus, and the process can be initiated with other bacterium of the same kind. But as I told you before, that this particular conjugation, it does not result in any recombination. All that happens is that this particular fertility factor or F factor, it has been transferred from one bacterium to another, and that's all. The original chromosome of both the bacteria, they are untouched, and no genes have been transferred, or there is no net gain or loss of genes on either side in both the bacteria. So this was the conjugation process in which F factor gets transferred from one cell to another, but there is no recombination. However, there are other varieties of this process in which actual recombination takes place, and that is what we are going to see in the next video. Thanks for watching, and see you in the next video.